welcome to G Fun Facts Online. In every liter of seawater, there are secrets, but one of the most enduring is a tiny creature found everywhere, from the sweltering tropics to the frozen poles. For more than 140 years, we've only known its baby picture. Its adult form is a complete phantom, seemingly vanished from the Earth. This is the century-old riddle of the Y-larva, a zoological detective story that takes us from 19th century research ships to the cutting edge of genomic science. The story begins in the late 1800s, a golden age of oceanic exploration. As scientists dragged fine meshed nets through the water, they pulled up a bewildering microscopic menagerie. In 1887, German zoologist Christian Hensen first collected these strange little creatures, but misidentified them. The real mystery began in 1899 with the Danish crustacean expert Hans Jacob Hansen. He recognized these larvae as something totally unique. He noted their distinctive, shield-like head, covered in intricate plates that looked like facets. He couldn't match this larva to any known adult crustacean, but it did look a bit like a baby barnacle. Unable to place it, he gave it a provisional name that would stick for over a century, Nauplius Y, with the Y simply signifying unknown. Thus, the Y-larva was born, a name perfectly capturing its enigmatic status. For decades, it remained just that, a widespread but perplexing presence. In 1985, the group was formally named Facetotecta, meaning faceted roof, a direct reference to that unique armored head. But this created a bizarre situation in the world of zoology. The Facetotecta became the only group of crustaceans whose entire classification was based solely on its larval forms. The adult remained a complete ghost. To solve the riddle, detectives had to study the clues, and the clues were the larvae themselves. The life of a Facetotectan, as we know it, unfolds in two distinct stages. It begins as a Y-Nauplius. This is the earliest free-swimming larval stage, measuring a mere 250 to 620 micrometers, about the width of a few human hairs. Under a microscope, it's a striking creature. Its most prominent feature is that dorsal head shield, the faceted roof that gives the group its name, covered in a complex pattern of armor-like plates. It has a single, simple eye at the front and propels itself through the water using 3 pairs of swimming appendages. The Y-Nauplius passes through a series of molts, shedding its exoskeleton to grow, before it undergoes its first major transformation. After its final naupliar molts, the Y-larva metamorphoses into its second larval form, the Y-cyprid. The discovery of this stage was a major step forward, because the cyprid is a hallmark of the Thecostraca, the major group of crustaceans that includes barnacles. This confirmed the Y-larva was indeed a barnacle relative. The Y-cyprid is a non-feeding stage, built for a single crucial purpose: to find a place to settle down and begin its adult life. It's more complex than the Nauplius, with a bivalved carapace, large compound eyes, and special antennules equipped with adhesive glands to explore surfaces and glue itself down. It's essentially a biological homing missile. But for years, this is where the trail went cold. Scientists could collect both Nauplii and cyprids from the ocean, but no one had ever seen a cyprid settle. They tried raising them in labs, offering all sorts of surfaces to attach to rocks, shells, and other things barnacles love. But the Y-cyprids would just swim until they exhausted their energy reserves and died. The crucial trigger for the final transformation was missing. The ocean was full of these questing larvae, but their destination was a complete mystery. This frustrating dead end led to a powerful and tantalizing hypothesis. What if the adults were hidden in plain sight, living a life so radically different that no one recognized them? The leading theory was that they were endoparasites, living their entire adult lives inside the bodies of other marine animals. This would explain why they were never found on rocks or in the sediment. If a Y-cyprid's job was not to find a rock, but to find a specific living host, its refusal to settle in a sterile laboratory dish suddenly made perfect sense. This idea gained huge traction from comparisons to another bizarre group of barnacle relatives, the Rhizocephala. These are infamous parasitic castrators. A female rhizocephalan cyprid attaches to a host, usually a crab, and injects a small cluster of cells into its body. This cluster grows into a root-like network, absorbing nutrients and hijacking the crab's hormones. The parasite castrates the host and forces it to care for the parasite's egg sac as if it were its own. Could the Y-larvae be doing something just as insidious? The smoking gun came in a landmark 2008 experiment. A team led by Henrik Glenner and Jens Hoeg, working with Y-larvae from Japan, decided to test the endoparasite hypothesis directly. Knowing that hormones trigger molting in crustaceans, they exposed captive Y cyprids to a synthetic version of the crustacean molting hormone, 20-hydroxyecdysone. The result was nothing short of mind-blowing. The hormone treatment worked. It triggered the Y cyprids to undergo the next stage of their life cycle, a metamorphosis no one had ever witnessed. But what emerged was not a tiny barnacle. Crawling out of the empty shell of the cyprid was a creature that defied all expectations. It was a slug-like, worm-like organism, entirely unsegmented and lacking any limbs or other recognizable arthropod features. It had no gut, and only the disorganized remnants of the cyprid's compound eyes remained as pigmented spots. The scientists named this new stage the ypsigon, a combination of Y-larva and vermigon, the name for the invasive stage of those parasitic rhizocephala. The ypsigon was capable of vigorous wriggling motions, as if it were designed to burrow into soft tissue. This discovery was the most significant breakthrough in the century-long riddle. It was not the adult, but it was the missing link, an additional larval stage perfectly adapted for invading a host. The implications were profound. The extreme anatomical simplification was a hallmark of an advanced endoparasite. By shedding its limbs, shell, and complex senses, it had become a living injection system, a biological hypodermic needle designed to deliver its cells into an unsuspecting host. The mystery of why adult Y-larvae had never been found was suddenly clear. Scientists had been looking in the wrong place. The adults weren't on the seafloor. They were almost certainly inside another animal. The striking similarity between the Facetotectan ypsigon and the rhizocephalan vermigon immediately raised a critical evolutionary question. Did this mean they were each other's closest relatives, having inherited this parasitic strategy from a common ancestor? For a time, this seemed plausible. However, as scientists began to apply the tools of molecular phylogenetics, analyzing DNA to reconstruct evolutionary family trees, a more complex and even more fascinating picture began to emerge. A major study published in 2025 in the journal Current Biology involved sequencing the entire transcriptome, all expressed RNA molecules, from over 3,000 Y-larvae. This big data approach provided a massive amount of genetic information to build a robust tree of life for barnacles. The results were definitive. The analysis confirmed that Facetotecta are not closely related to the parasitic Rhizocephala at all. Instead, they occupy a distinct, early branching position on the family tree. This means that the astoundingly similar parasitic life strategy, a free-swimming larva that transforms into a limbless, worm-like invasive stage, evolved not once, but twice, independently, in two separate lineages. This is a textbook example of convergent evolution, where unrelated organisms independently evolve similar traits to adapt to similar challenges. The evolution of wings in birds, bats, and insects is a classic example. The parallel evolution of the ypsigon and the vermigon is one of the most remarkable cases of convergence ever documented. The intense selective pressure of a parasitic lifestyle was so powerful that it forced two different lineages to abandon their complex arthropod bodies and arrive at the exact same, elegantly simple solution: a living delivery system for a parasitic payload. As if that tale of convergent evolution wasn't surprising enough, The recent wave of genetic research has uncovered another secret about Y-larvae: their incredible and almost entirely undocumented biodiversity. For over a century, the formal taxonomy of Facetotecta had been stagnant. Only about 17 species had been officially named. This paltry number stood in stark contrast to the larvae's widespread distribution. The genetic data blew this picture wide open. The 2025 transcriptome study, along with other molecular surveys, revealed a staggering level of hidden diversity. In a single harbor in Japan, researchers identified over 100 genetically distinct species of Y-larvae. This suggests that the true number of Facetotectan species worldwide could be in the hundreds or even thousands. The 17 named species are just the tip of a colossal iceberg of biodiversity that has remained completely invisible to science. This discovery means that the Facetotecta are not a minor, obscure group of crustaceans, but a major and ecologically significant one. If each of these hundreds of species is a parasite, they could be playing a crucial, yet completely unappreciated role in marine ecosystems. They could be controlling host populations, influencing food webs, and driving evolution, just like their parasitic barnacle cousins do. A vast, hidden army of Facetotecta could be quietly shaping the dynamics of life beneath the waves. So despite these monumental leaps forward, the original century-old riddle remains partially unsolved. We now have an incredibly strong suspect, an endoparasite, and we have the getaway vehicle, the ypsigon. But the adult itself remains at large, and its hideout, the host, is still a mystery. Finding them presents a monumental challenge. The adult is likely a highly reduced, amorphous blob or root-like network, bearing no resemblance to a crustacean, hidden deep within the tissues of another animal. The search area is, quite literally, the entire animal kingdom of the sea. Modern molecular techniques offer a targeted approach. Scientists are now developing methods to screen potential host organisms for Facetotectan DNA. This involves collecting tissue samples from a wide range of marine invertebrates, crabs, shrimps, worms, mollusks, and using highly specific genetic probes to search for the Y-larva's unique genetic signature. It is a painstaking process of elimination, but it represents the most promising path to finally identifying the host. The day that a scientist finally matches a strand of Facetotectan DNA to its host organism, the last piece of this epic puzzle will click into place, and one of marine biology's most enduring mysteries will finally be solved. Until then, the Y-larva continues its silent, enigmatic drift through the world's oceans, a constant reminder of the boundless secrets the sea still holds.